Alright, so in this lecture we're going to cover the structure and function of macromolecules. Macro means large, so when we're talking about macromolecules we're going to be dealing with large molecules like carbohydrates, lipids, proteins, and nucleic acids. And one thing we need to talk about up front is the term macromolecule versus polymer. So macro means large molecule, poly means many, and polymers are large molecules. So polymers are macromolecules, but not all macromolecules are going to be polymers. So for example, complex carbohydrates, proteins, and nucleic acids would be considered macromolecules, but they'd also be considered polymers because they're going to be made up of many smaller subunits. Whereas something like a lipid would be a macromolecule because they are large, but it does not meet the definition of polymer because it is not going to be made up of repeating units. When we talk about each of the organic compounds, we will see why they are either macromolecules and polymers or just macromolecules. So let's go ahead and start talking about carbohydrates first. Carbo means carbon, hydrate, water. So if you take a look at the general formula, you see carbon and water, and the hydrogen to oxygen ratio is going to be in a ratio of 2 to 1, just like water. This general formula for carbohydrates means that the empirical formula for a monosaccharide is CH2O. It's the reduced form of a monosaccharide, which the general formula for a monosaccharide would be C6H12O6. But then we have this N on the outside. So what that number means, it's going to be the number of monosaccharides that are put together to form a disaccharide or a polysaccharide. Now we're talking about carbohydrates. We're also talking about things that are going to be used as energy and building blocks for other things. So carbohydrates, monosaccharides specifically, are going to be monomers, mono meaning single. So we're going to put a bunch of single units together, like glucose, in order to form our larger disaccharides or polysaccharides. Something else you want to take a look at is that glucose, a simple sugar, sucrose, a more complex sugar, they both end in OSE. And so you'll note that OSE is a sugar ending. Now, if we want to put these together, we're going to do this through a process called dehydration synthesis. Okay, dehydration synthesis, you're going to lose a molecule of water in synthesizing the larger molecule. You could break them down by adding water back in, and that would be called hydrolysis. But when you put a sugar combined to another substance, it's going to form something called a glycosidic linkage. And so you can think of glyco like gluco like sugar. So this is going to be a sugar linkage. So when we take two monosaccharides and we put them together to form a disaccharide through the process of dehydration synthesis, we get something like this, sucrose here, which would actually be binding a glucose and a fructose molecule. And we get the formula C12H22O11. But if you were to actually add C6H12O6 plus C6H12O6, you would actually get C12H24O12. So where did the other two hydrogens and the oxygen go? That's the water that is released as a result of dehydration synthesis. So now if we're going to take some of these disaccharides and we're going to continue to build upon them, we form something called a polysaccharide, poly meaning many. Now these polysaccharides can function as energy storage compounds, okay, so glycogen in animals, okay, and starch in plants, or structural compounds like cellulose. Again, note the OSE ending. Cellulose is going to be found in the cell walls of plants, okay, and the difference between cellulose and starch is going to be due to the difference in the form of glucose used. So we're going to be using an alpha form, this A stands for the A for alpha, or a B for beta form of glucose. And then we have structural compounds also found in animals, and that's going to make up chitin, which is going to be monomers of glucose, which have a nitrogen group attached to it. All right, so moving on into lipids. So lipids, these are going to be things like fats, waxes, and oils. We have our groups. We've got triglycerides. Think of the fats and waxes and the oils that we associate with that. We have phospholipids, and we have sterols. Now, for the triglycerides, they're going to be composed of two parts, glycerol, and then 3 fatty acids. And once we use those, we are done. We have produced our lipid. So this is why this is not considered a polymer, because we don't continue to build glycerol and fatty acids in a chain. Glycerol, 3 fatty acids, we've made our triglyceride, it's done. Okay? Glycerol is a 3-carbon alcohol. It's got the formula C3H8O3. And then the 3 fatty acid chains, okay, they are simply long chains of hydrocarbons, so carbon chains with a bunch of hydrogens added onto them, as well as a carboxyl group on one end. Okay, so these 3 fatty acids are going to store energy in their carbon to hydrogen bonds, and this is why these particular molecules have lots and lots of energy, because they have such a large number of carbon to hydrogen bonds. So these large hydrocarbon chains, these fatty acid tails, can be saturated or unsaturated. And all this simply means is unsaturated is not holding as many hydrogens as it could. That means that there are going to be double bonds between carbons. Saturated means it's holding all the hydrogens that it can, and that means that there are no double bonds between the carbons. Now, how you can tell the difference between them looking at them, if you had them in front of you, is that unsaturated is going to be liquid at room temperature, whereas saturated fats tend to be solid at room temperature. So think like lard or Crisco, and then this would be, say, corn oil or something of that nature, cooking oil, olive oil, something like that. So these hydrocarbon chains are going to form a hydrophobic tail. Okay, that hydrophobic tail means that it does not like water, and we're going to talk a little bit more about hydrophobic in a moment. So let's go on to the second class of lipids, phospholipids. These are going to have a phosphate group, okay, as well as 2 fatty acid tails and our glycerol. So the difference between a phospholipid and a triglyceride is we are going to exchange 1 fatty acid for a phosphate group. Okay, now this phosphate group is going to have a hydrophilic end. It's going to like water. All right, and so what this is going to do, this hydrophobic tail and this hydrophilic head, is going to help to create our bilayers, our phospholipid bilayers, or our cell membranes or plasma membranes. And in this case, the hydrophilic heads are going to face outwards, okay, out into the environment or inside into the cell. And in between, you're going to have your double layer of hydrophobic tails, okay, creating this phospholipid bilayer. Third group we have are sterols. So these can function as chemical messengers, and the characteristic about these you'll note is that they are molecules that have 4 carbon rings attached with various groups attached to those, which would lead to the different functions that they would exhibit. So one last thing to remember before we move on talking about the nucleic acids is the bonds that are going to be produced when we bind our glycerol to our fatty acids are called ester bonds.